Hi there, my name is Kirsten Bryant, and I'm an assistant professor in the Department of Pharmacology at the University of North Carolina at Chapel Hill. I'd like to start off by thanking the organizers for inviting me to speak today, and I'm excited to share some of our science with you. Today I'm going to be speaking to you about inhibiting the RAS pathway as a treatment approach for pancreatic cancer. I wanted to start with a brief introduction to this pathway. There are three RAS isoforms, KRAS, NRAS, and HRAS. As a family, these mutations in these proteins drive about 25% of human cancers. KRAS is mutated primarily in pancreatic cancer, and that will be the topic of our discussion today. The pathway downstream of KRAS that I will focus on is the RAF-MEK-ERK pathway. Some of the cancers with the highest frequencies of RAS mutations are pancreatic, colorectal, and lung adenocarcinoma. And when we look at cancer-associated deaths in the US, these three cancers are at the top of the list. Therefore, there is clearly a clinical need to target this pathway. Pancreatic cancer has the highest frequency of RAS mutations. Therefore, our lab is interested in targeting the RAS pathway for pancreatic cancer treatment. And because compounds that bind directly to RAS have been hard to develop, we have focused our efforts on targeting pathways downstream of RAS, many of which are pictured here. However, if you follow the field, you may be thinking to yourself that we actually do have direct RAS inhibitors now. And this is true. Sotorasib, the first RAS inhibitor, was approved by the FDA in June 2021. However, this inhibitor and others that have been developed specifically target mutant RAS in which the hotspot glycine 12 has been mutated to a cysteine. While KRAS G12C mutations make up 14% of the RAS mutations in all human cancers, this frequency comes primarily from lung adenocarcinoma, where 30% of patients harbor a RAS mutation, and of those 30%, 52% are KRAS G12C mutations. Unfortunately, only 2% of KRAS mutations in PDAC are G12C mutations. Therefore, for this particular cancer, our best bet is still targeting the pathways that are downstream of RAS. So today I'm going to begin my talk by discussing some published work on how three of these strategies converge on the process of autophagy. Then I'm going to transition into ways that we are endeavoring to improve dual ERK MAPK kinase and autophagy inhibition for pancreatic cancer treatment. It's now well accepted that mutant KRAS alters the metabolic state of PDAC. Mutant KRAS has been linked to upregulated glycolysis, differential channeling of glutamine intermediates and glycolytic intermediates, and it's also been associated with a differential utilization of nutrient scavenging pathways, primarily autophagy and macropinocytosis. Today, I am going to focus on the metabolic process of autophagy, which is literally cellular self-eating during times of nutrient stress. Autophagy has been implicated in sustaining the tumorigenic growth of pancreatic cancer. Pancreatic cancer primary tumors and cell lines have elevated autophagy under basal conditions. And inhibition of the autophagy pathway, either genetically or pharmacologically, has been shown to cause tumor regression and prolonged survival in pancreatic cancer xenografts and genetic mouse models of PDAC. This was the state of the field when I entered it in about 2014. Coming from a RAS lab, we had a very simple initial hypothesis. Because so many pancreatic cancers are driven by RAS mutations, and because autophagy is upregulated in pancreatic cancer, we started with the very simple hypothesis that KRAS is what was driving the high levels of autophagy in PDAC. And our first experiment was to simply knock down KRAS with the hypothesis that we would see a loss in autophagy. However, what we observed was the exact opposite. we actually saw that silencing of KRAS increases glycolytic flux. I'm going to be using this autophagy reporter system a lot throughout my talk, so I'm just going to take a few seconds here to describe it. What we're overexpressing in the cells is an mCherry and EGFP tagged LC3B. While this construct is localized to the autophagosome, both the mCherry and EGFP fluorescence is observable. However, when the autophagosome fuses with the lysosome, there's a quenching of the EGFP fluorescence, and you're left with just the mCherry fluorescence. Therefore, you can read out a ratio of mCherry to EGFP to read out autophagic flux. A higher ratio, meaning faster flux through the pathway. So as I mentioned, the first thing we did was we knocked down KRAS, and I'm showing here that following genetic silencing in a panel of pancreatic cancer cell lines, we saw increase in autophagic flux when we genetically silenced KRAS. We do have one KRAS G12C model available in terms of cell lines, and when we treated the MEKI-PIKfyvei cells with the KRAS G12C inhibitor, we also saw an increase in autophagic flux. And finally, using an inducible KRAS mouse model developed by the DePinto group, we were able to show that loss of KRAS in cell lines derived from this mouse model increased autophagic flux as well. However, as I mentioned, we do not have KRAS inhibitors that can be used for many PDAC patients. So therefore, we looked further downstream in the pathway at a node that could be targeted. And so what we focused on was ERK inhibition. And here what I'm doing is showing you that ERK inhibition phenocopies KRAS loss in that it increases autophagic flux in both our panel of human PDAC lines as well as this inducible mouse model. So therefore, we quickly disproved our original hypothesis. But this observation that ERK inhibition or KRAS loss led to an increase in autophagic flux made us hypothesize that perhaps the cells were becoming more dependent on autophagy in this state. And therefore, that concurrent inhibition of both ERK and autophagy may be a good treatment strategy. And summarizing a lot of work, I'm going to tell you that this is indeed a synergistic relationship. Looking first at human cell lines, we showed that the addition of an autophagy inhibitor, chloroquine, shifted the dose response curve to an ERK inhibitor. We observed this in a panel of PDAC cell lines across a range of different ERK inhibitor concentrations. Using combination index as an indicator of synergy, we see a synergistic relationship across a broad range of concentrations. We transitioned these studies into organoid models, and the synergy was maintained. And no matter what node of the pathway we inhibited, whether it be ERK or MEK or RAF, we continued to see a synergy between ERK MAP kinase inhibition and autophagy inhibition with chloroquine. Finally, when we extended these studies into a PDX-derived mouse model of PDAC, again, we saw that tumor weights were decreased with the combination more so than with either agent alone, and survival was enhanced. At the time that we published these results, we co-published with another group that had focused on MEK inhibition, whereas we had focused on ERK inhibition. And another paper came out the same month, focused on RAF inhibition. All three papers showing that inhibition of each node of the MAP kinase pathway synergized with autophagy inhibition. And this has led to somewhat of a resurgence in interest in autophagy inhibition as a PDAC treatment. This is being tested in a variety of clinical trials right now, some led by the first author of the companion paper that came out with ours. And we're very pleased to be involved in two of these trials as well, one of which is being performed here at UNC, a combination of ERK inhibition with autophagy inhibition. So while we're very eager to see the outcome of these trials, we still know that there is much to be improved with this combination. And therefore, the focus of my lab right now is understanding ways in which we can improve upon dual MAP kinase and autophagy inhibition. So the first story that I'd like to tell you about it today is a way that we're interested in identifying new or additional sensitizers to autophagy inhibition or identifying better autophagy inhibitors. So the first story that I'm going to be talking about today is focusing on additional sensitizers. To address this question, we performed a CRISPR-Cas9 screen using a library loosely regarded as the druggable genome. So this encompasses about 2,200 genes that we have therapeutic agents against. And we performed this screen in the presence and absence of the autophagy inhibitor chloroquine, with the idea being that we would identify additional sensitizers to chloroquine treatment. We used the MAGIC analysis package to quantify our sgRNA abundances for each gene at the end of the screen. And then we performed GSEA analysis to rank the MAGIC beta scores. We identified the hallmark mTOR-C1 gene set among the most significantly depleted gene sets. And this was a comforting result to observe, as others have shown that mTOR inhibition enhances metabolic reliance on autophagy. We found that multiple genes within the mTOR pathway were depleted in chloroquine-treated cells, and one of the most depleted genes that we observed was the IGF receptor. As an orthogonal approach to characterizing the effect of chloroquine treatment on PDAC cells, we also sought to characterize the signaling changes that accompany chloroquine treatment by reverse phase protein array, or RPPA analyses. These can be thought of as a large Western blot where we can look at the signaling status of about 250 different proteins all at once. So these are some of our top hits. Consistent with the mechanism of chloroquine prohibiting autophagosome degradation, chloroquine treatment caused a dramatic increase in the autophagosome-associated LC3B protein, as well as the lysosomal protein LAMP2. This was a comforting control to see. Interestingly, chloroquine treatment also caused a marked increase in phosphorylation and activation of receptor tyrosine kinases, including the IGF receptor. When we utilized our CRISPR screening data to investigate whether the loss of any of these RTKs or other signaling proteins were sensitizers, we found that the IGF receptor to be selective, in that it was both a hit in our CRISPR screen as well as upregulated in the RPPA analysis. And we were able to validate this result in-house. So you're looking here at a Western blot, and you can see that upon chloroquine treatment, we see an upregulation in the phosphorylated state of the IGF receptor, whereas we don't see changes in total IGF receptor. To validate this result, we performed dose-response studies in the presence of an IGF receptor inhibitor as well as chloroquine. So you see the IGF receptor dose response across the bottom, and then we're stepping in different concentrations of chloroquine. And again, this was also a synergistic relationship. We also observed that treatment with an IGF receptor inhibitor led to an increase in autophagic flux, not to the same degree as what we observed with ERK inhibition, but to a significant degree. And then in collaboration with Stephen Hersting's lab here at UNC, we performed a mouse model study in which we looked at the combination of chloroquine and our IGF receptor inhibitor and saw a significant decrease in tumor growth in the dual treated mice. However, while this change was significant, statistically, we still had fairly large tumors remaining in the combination treatment. Therefore, we went about trying to understand how we could further push the reliance of IGF receptor inhibited cells on autophagy. To do this, we selected a panel of 11 clinically relevant inhibitors and also included inhibitors that were previously established to modulate autophagy. We then performed a small-scale fluorescence-based assay that utilized our PDAC cells overexpressing our autophagy reporter construct, and we used flow cytometry to monitor autophagic flux. So what we are looking at here is the effect of each of these inhibitors alone, and then when we layer on the IGF receptor inhibitor. And what you can see is MEK and ERK inhibitors stand out as agents that can further induce autophagy when combined with an IGF receptor inhibitor. And that's quantified here. Here you can see a direct comparison of the IGF receptor inhibitor in orange to an ERK inhibitor in blue. So you can see that while an ERK inhibitor can induce more autophagic flux than an IGF receptor inhibitor, the combination treatment shown in purple here has the highest level of autophagic flux induced. So IGF receptor inhibition can further enhance autophagy in an ERK inhibitor state. Looking at upstream signaling, primarily phosphorylated AMPK, you can see that both IGF receptor inhibition alone and ERK inhibition alone cause an increase in phosphorylated AMPK, but it's the combination that can drive an even greater increase at a range of different concentrations. Interestingly, we also showed that this combination was more cytotoxic. So here I'm showing spheroid assays where we're reading out apoptosis. The matrix is of an ERK inhibitor and IGF receptor inhibitor, and you can see the panels of minus and plus chloroquine, and appreciate that there is more cell tox fluorescence when we add on chloroquine to this combination. This is reproduced in a number of different pancreatic cancer cell lines. And finally, we moved these studies into organoid cultures and showed again that the triple combination of ERK inhibition, IGF receptor inhibition, and chloroquine dramatically reduced PDAC organoid growth. And so therefore, we conclude that the addition of an IGF receptor inhibitor further potentiates the ERK chloroquine combo. Moving forward, we're interested in further validating this combo in more in vivo models, this triple combination, I should say, in vivo models. And then we're also interested in the dramatic upregulation of RTK signaling in general upon chloroquine treatment. When cells are treated with chloroquine, there is a large increase of autophagosomes in the cell. And we know that the receptor tyrosine kinases are endocytosed following activation. And some literature suggests that these receptor tyrosine kinases are still capable of signaling from endosomes. And therefore, we're interested in determining whether receptor tyrosine kinases are capable of signaling from internalized structures following chloroquine treatment. The last thing that I'd like to talk to you about, the last thing I would like to speak about today is our efforts to identify better inhibitors of the autophagy pathway. So hydroxychloroquine is not a particularly potent autophagy inhibitor, and it's not specific to the autophagic pathway. And therefore, we hypothesized that if we could determine a better way to more potently inhibit this pathway, the combination in general would be more durable. So going back to our druggable genome CRISPR screen and focusing now on our DMSO treated cells, while this library was not designed to be focused on the autophagy pathway, there were a number of autophagy related genes that happen to be part of this library, approximately 70. And of the 70 autophagy related genes, roughly half of them hit as sensitizers in the screen. And that just further enforces the finding of others that PDAC is reliant on autophagy for growth. We were particularly interested in this lipid kinase PIK5, and that's what we're going to be talking about for the rest of the talk. Focusing a little bit more on the details of the autophagy pathway, chloroquine, which we've spent most of this talk focused on, is a general inhibitor of the ability of the lysosome to fuse with autophagosomes, and therefore an end of the pathway inhibitor. However, there are a variety of different upstream events that can also be targeted. I won't have time to talk about these today, but this is another focus of our laboratory. So autophagy is initiated upon ULK kinase activation in form of this green initiation complex shown here. And then this activates a nucleation complex shown in blue. And that forms an isolation membrane. And this is where LC3, which we've discussed earlier, gets incorporated into the autophagosome as it forms. And eventually the autophagosome fuses with the lysosome. And then this is what causes degradation and recycling of materials. PIK5 can be inhibited by the compound apilimod. And working with SpringWorks Therapeutics, we became interested in understanding the effects of apilimod on the autophagy pathway. So just a little bit of background on PIK5. PIK5 is a lipid kinase that's responsible for converting PI3P to PI35P2. It is through this enzyme that PI35P2 is made. This is the gatekeeping enzyme for making PI35P2. While it's appreciated that mitochondria fuse and fizz quite dynamically in the cell, you may not know that lysosomes actually do the same thing. So they're very dynamic organelles, and they can both fuse together and then also separate. And PIK5 is important for this ability of lysosomes to separate. So therefore, when PIK5 is inhibited, lysosomes will become enlarged in the cell. Also interesting to us as a potential target for cancer treatment, PIK5 is not an essential gene as defined by DepMap. So it would not be a generally toxic target. When we treat our PDAC cells with a PIK5 inhibitor, we observe a very dramatic phenotype, as you might expect from that last cartoon. So as the cells are treated with increasing concentration of this PIK5 inhibitor, apilimod, we start to see this large vacuolization of the cells, which is very evident here in the 300 nanomolar. And what these vacuoles are these enlarged lysosomes. Interestingly, the appearance of these vacuoles in the cell correlates perfectly with the GI50 for apilimod. We also see that inhibition of PIK5 results in an inhibition of autophagy. So what we're looking at here are some common markers of autophagy. P62 is a general cargo protein, and we've talked about LC3B. The cytosolic form of this protein is LC3B1, and LC3B2 is the autophagosome-associated isoform. So what I'm showing you here is following 24 hours of apilimod treatment, we see both an accumulation of P62 and an accumulation of LC3B2 across our dose response of PIK5 inhibitor. This inhibition is maintained following days of treatment, actually. So here the inhibitor is replenished every 3 to 4 days, and we see that autophagy is still robustly inhibited at these longer time points. So while we are interested in identifying additional autophagy inhibitors, we certainly feel that for pancreatic cancer treatment, we will always have to pair an autophagy inhibitor with a MAP kinase inhibitor. So we next decided to determine whether or not we would see synergy between a ERK MAP kinase inhibitor and a PIK5 inhibitor. So here we're looking at the MEK inhibitor, mirdametinib, and the PIK5 inhibitor, apilimod. The dose response for the MEK inhibitor is shown, and then each color is an increasing dose of apilimod. And you can see here that we see a dramatic shift in the GI50 when we're treating with increasing doses of apilamod, and we're quantifying synergy below in the heat maps using BLISS, red being a synergistic interaction. So you can see across a variety of PDAC cell lines, across a large matrix of concentrations, we see a synergistic relationship between MEK inhibition and apilamod in treatment. This relationship is maintained when we move from our 2D cell culture models to our organoid models here. We see dramatic synergy in our organoid models as well. So because PIK5 has such a dramatic effect on cellular morphology, we were somewhat concerned about toxicity of this. So therefore, we looked at the effect of PIK5 inhibition on our non-transformed normal pancreatic cancer cell lines, HPNE. Of course, they have to be somewhat transformed to use in culture, but they're the most normal model that we have. As you can see here in blue, we do not see a dramatic effect of apilamod treatment on the growth of the HPNE cells. We have HPNE cells that are transformed with just the RAS oncogene. And you can see that upon transformation with RAS, we do see a shift in the curve. And this makes us believe that perhaps this will be specific to our transformed cancer cases. One of the reasons we were particularly excited about this result is this is not something that we typically see with MAP kinase inhibitors. So here you can see an ERK inhibitor is quite toxic to both HPNE and HPNE RAS cells. And the same is true generally to MEK inhibitors. However, we do see the largest separation that we've observed to date using mirdametinib, more so than what we see with trametinib, which at least at this point makes us quite pleased at the potential therapeutic window for this combination. One of the last points that I'd like to tell you about is that the combination of ERK MAP kinase inhibitors with PIK5 inhibitors appears to be a more apoptotic combination. So I'm showing you an Annexin 5 PITC assay here, and you can appreciate that both MEK and PIK5 inhibition does not induce a large amount of apoptosis. However, when you look at the combination of the two, then you start seeing a dramatic increase in the levels of apoptotic cells. And this is maintained in multiple cell lines. And when we compare this to the results that we've had in the past with chloroquine, you can see that whereas we do see somewhat of an increase in apoptosis in the chloroquine-treated cells, so those are the ones towards the far right of the graphs, the combination of PIK5 inhibitors with MEK inhibitors is a much more apoptotic combination. So we're very interested in trying to understand why this is the case. And we have some preliminary data that's starting to shed some light on this. What we did was we performed an apoptosis array in DMSO-treated cells, each agent alone, and then the combination. And what we observe is a loss of anti-apoptotic proteins in the combination treatment. Additionally, we see an increase in P27-KIP with the combination, more so than either agent alone. And this is indicative of a G1 arrest. And so we're currently looking more in depth into the effects of this combination on the cell cycle. So what I've shown you in this part of the talk is that PIK5 inhibition inhibits autophagic flux, and it also synergizes with MEK inhibition. And moving forward, we're interested in further characterizing these differential effects of PIK5 inhibition and chloroquine. And so one of the ways that we've gone about doing this is to submit samples for RPPA analysis in collaboration with Chip Petricoin at George Mason University. And therefore, by casting this wide net, we hope to observe which pathways are differentially regulated. We're also interested in moving our validation of this combination from organoids now into in vivo mouse models of PDAC as well. So the main conclusions that I've presented to you today is first our original observation that ERK MAP kinase inhibition causes pancreatic cancer cells to become addicted to autophagy, and that this combination is a potential therapeutic approach for KRAS mutant PDAC. We furthered this story quite recently by showing that IGF receptor inhibition further induces ERK inhibitor cells' reliance on autophagy, and this triple combination is more potently cytotoxic in PDAC. And then finally, looking at different ways to inhibit the autophagy pathway, I have presented PIK5 as a potential target and shown that this autophagy inhibition strategy also synergizes with ERK MAP kinase inhibition. With that, I'd like to acknowledge the people who do all the work. So this is my lab. The PIK5 story that I presented is one of the projects of my first graduate student, Jonathan DeLiberty. And he's been helped quite a bit by our technician, Noah Pieper, and Runying Yang, our organoid specialist. The IGF receptor story was driven by a postdoc in Channing Durr's lab. They're right next door, Clint Stallnecker. And I'd also like to acknowledge my other collaborators in funding. Thank you so much for your attention today.